Impurity qualification is one of the most discussed topics in the pharmaceutical industry. Let us understand the concept of ICH M7 guideline related to assessment and control of DNA reactive mutagenic impurities in pharmaceuticals to limit potential carcinogenic risk. What is this ICH M7R1? ICH is the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. ICH topics are divided into 4 categories: Q. Quality Guidelines, S. Safety Guidelines, E. Efficacy Guidelines, and M. Multidisciplinary Guidelines. R denotes revision number. Why the ICH M7 guideline? It is to limit the potential carcinogenic risk related to impurities. How does the guideline limit the potential carcinogenic risk related to impurities? It is by identifying, categorizing, and controlling the levels of impurities. What is the type of impurity that is to be controlled? They are DNA reactive or mutagenic impurities. In what? In pharmaceutical products. How to identify? Computational toxicology assessment is used to identify the mutagenicity, which predicts the outcome of a bacterial mutagenicity assay. 2 quantitative structure-activity relationship prediction methodologies should be used to predict the mutagenicity. One methodology used should be rule-based and another should be statistical-based. How are these impurities categorized? Class 1, Class 2, Class 3, Class 4, Class 5. Class 1. Known mutagenic carcinogens come under Class 1. In Class 2, it is known mutagens with unknown carcinogenic potential. For example, bacterial mutagenicity is positive, whereas no rodent carcinogenicity data is available. Under Class 3, it is the impurity contains alerting structure which is unrelated to the structure of the drug substance and no mutagenicity data is available. In Class 4, the impurity contains alerting structure and the same alert in the drug substance or compounds related to the drug substance. Example, process intermediates, which have been tested and found to be non-mutagenic. The Class 5 impurity contains no structural alerts or alerting structure with sufficient data to demonstrate lack of mutagenicity or carcinogenicity. How impurities are controlled? For Class 1, it is to be controlled at or below compound-specific acceptable limit. For Class 2, it is to be controlled at or below acceptable limits. Appropriate TTC, that is, threshold of toxicological concern. A TTC-based acceptable intake of a mutagenic impurity is 1.5 microgram per person per day. For Class 3, it is to be controlled at or below acceptable limits. appropriate TTC or conduct bacterial mutagenicity assay. If bacterial mutagenicity assay proves it non-mutagenic, it will be classified as class 5, and if mutagenic, as class 2. For class 4, treat as non-mutagenic impurity. For class 5, treat as non-mutagenic impurity.